Cell, the unit of life chapter. Quick revision. Cell is a fundamentally structural and functional unit of all living organisms. Anything less than a complete structure of the cell does not ensure independently living. Anthony van Leeuwenhoek first saw and described the life cell. Before him, Robert Hooke discovered the cell, but that was a dead cell. In 1838, a German botanist examined large number of the plants and observed that all plants are composed of the different kinds of the cells which form the tissues of the plant. At the same time, in 1839, a British geologist reported that animals had the thin layer which is known as a plasma membrane. He also concluded, based on his studies on the plant cell, plants have the cell wall. Sleeden and Swan together formulated the cell theory, but this theory did not explain how the new cells are formed and divided. Later, in 1855, Rudolf Virchow explained that new cells arises from the pre-existing cells, hominous cellula e cellula, and finally modified the cell theory as all living organisms are composed of the cells and product of the cells. All cells arises from the pre-existing cells. Cells differs greatly in shape, size and activities. For example, mycoplasma is the smallest cell, while the egg of an ostrich is the largest isolated single cell. Nerve cells are the sum of the longest cells. The cytoplasm is the main area for the cellular activities in both plants and animal cells. Ribosomes are the non-membrane bounded organelles which is found in both eukaryotic and prokaryotic. Apart from the cytoplasm, they are also present in the mitochondria, chloroplast and on rough endoplasmic reticulum. Animal cells contains another non-membrane bounded organelle called the centrosome which helps in the cell division. Cells that have the membrane bounded nuclei are called as the eukaryotic cells. Cells that lack the membrane bounded nuclei are called as the prokaryotic cells. Prokaryotic cells are represented by the bacteria, blue-green algae, mycoplasm, papillae. In addition to the genomic DNA, many bacteria have the small circular DNA outside the genomic DNA called the plasmid. Plasmid DNA confers certain unique phenotypic character to such bacteria. One such bacteria is resistance to the antibiotic. Plasmid DNA is used to monitor the bacterial transformation with the foreign DNA. All prokaryotic have the cell wall surrounding the cell membrane except the mycoplasm. Most prokaryotic cells have the cell envelope which is tightly bounded three-layered structure. The outermost glycocalyx and the middle one is cell wall and the innermost is a plasma membrane. Glycocalyx may be loose sheath called the slime layer, thick and tough called the capsule. The cell wall prevents the bacteria from bursting and collapsing. Extentations of the plasma membrane into the cell in the form of the vesicles, tubules and lamella are known as the mesosomes. Mesosomes also help in the cell wall formation, DNA replication, distribution of the daughter cells, respiration process, secretion and increase the surface area of the plasma membrane. In cyanobacteria, chromatophores contain the pigments. Each layer of the cell envelope performs distinct function. They act together as a single protective unit. The plasma membrane is selectively permeable in nature and interacts with the outside world. It is structurally similar to the eukaryotic. Bacterial may be motile or non-motile. If motile, they have the flagella composed of the three parts, filament, hook and basal body. Pilla and fimbriae do not play any role in the motility. Bacteria, on the basis of their difference in the cell envelope, can be classified into the two types, gram-positive and gram-negative. Ribosomes are the 70S, has the subunits 50S and 30S. Several ribosomes may attach to the single mRNA and form the chain called the polysome or the polyribosomes. Ribosomes are associated with the plasma membrane. The ribosomes or the polysome translate the mRNA into the proteins. Reserve material is stored in the form of the inclusion bodies in the prokaryotic cytoplasm. For example, phosphate granules, synoviac granules, and glycogen granules. Gas vacuoles are found in the blue, green, and purple and green photosynthetic bacteria. Beside the nucleus, eukaryotic cells have the other membrane-bounded structure which is known as the organelles like endoplasmic reticulum, Golgi complex, etc. The eukaryotic cells includes all protista, plants, animals and fungi. Plants have large vacuole and animal cells have the centriole which are absent in almost all plant cells. The eukaryotic ribosomes are the ATS in the cytoplasm are the smallest subunit 40S and larger ATS. Chemical studies on the cell membrane, especially in the human RBC, enabled scientists to deduce the possible structure of the plasma membrane. Cell membrane is mainly composed of the proteins and lipids. Lipids are mainly phospholipids that are arranged in the bilayer. Phospholipid contains a polar head which is outer side and non-polar tail hydrophobic which is towards the inner side. In human RBC, 52% is of protein, 40% is lipids. Membrane of protein can be integral and peripheral. Most accepted model for the structure of the cell membrane is fluid mosaic model was given by the Singer and Nicholson in 1972. Membrane is selectively permeable. Many molecules can move across the membrane without any requirement of energy is known as a poisson. Movement of the water by the diffusion is known as a osmosis. Many molecules can require the energy or ATP for their transport is called as the active transport. Example Na plus or K plus pump. The quasi-fluid nature of the lipids enable the lateral movement of the protein within overall bilayer. These ability to move within the membrane is measured as its fluidity. Polar molecules cannot pass through the non-polar lipid bilayer. The fluid nature of the membrane is important for the functions like cell growth, cell division, endocytosis, segregation and formation of the intercellular junctions. A non-living rigidity structure is known as a cell wall which form the outer covering of the plasma membrane. Is present in fungi and plants. Cell wall of the younger plant cell. The primary wall is capable of the growth which gradually diminishes as the cell mature and the secondary wall is formed and innerwards towards the membrane of the cell. The middle lamella is mainly made up of the calcium pentate. Algae have the cell wall made up of the cellulose, calcium, mannose and calcium carbonate. In plants, it consists of the cellulose, hemicellulose, pectins and proteins. While each of the membranous organelles is distinct in terms of its structure and functions, many of these are considered together as an endomembrane system because their functions are coordinated. Endoplasmic reticulum is a tiny tubular structure scattered in the cytoplasm which is known as the endoplasmic reticulum, nothing but ER. The ER which has ribosomes on the surface are called as a rough endoplasmic reticulum. In the absence of the ribosomes on the surface, they appear smooth, hence they are called as a smooth endoplasmic reticulum. Rough endoplasmic reticulum is involved in the protein synthesis, whereas a smooth endoplasmic reticulum is involved in the lipid synthesis. We all know that ribosomes' main function is the synthesis of the proteins. Ribosomes, where they present, they can synthesize only protein. In the absence of the ribosomes, they appear the smooth. It is involved in the lipid synthesis. Golgi apparatus were named Golgi bodies after the Camilo-Golgi. They consist of the cisternae which are concentratedly arranged near the nucleus with a distinct convex at the forming phase and concave trans or the maturing phase. Principally performing the function of the packing the material, it is important site for the formation of the glycoproteins and glycolipids. Lysosomes are the membrane-bounded vascular structure formed by the process of the packing in the Golgi apparatus. They are rich in the all hydrolytic enzymes, lipases, proteases, carbohydrates, optimally active at the acidic pH. These enzymes are capable of the digesting carbohydrates, proteins, lipids, and nucleic acids. Mitochondria usually, unless specifically stained, are not easily visible under microscope. Each mitochondria is a double membrane bounded structure with inner compartment called the matrix. The towards they have the two membranes and they have their own specific enzymes. The outer membrane forms the continuous limiting boundary of the organelles and inner membrane form the cristae. Mitochondria are the site for the aerobic respiration. They produce cellular energy in the form of the ATP, hence it is called as a powerhouse of the cell. The matrix has a single circular DNA molecules and a few RNA molecule ribosomes of the 70s and the components required for the synthesis of the proteins. Here as Sedberg's units stand for the sedimentation coefficient, which is indirectly measure the density and size. Mitochondria divided by the fusion. Plastids are found in all plants, cells and euglenoids. Based on the pigmentation, plastids can be classified into the chloroplast, chromoplast and leucoplast. Chloroplasts contain the chlorophyll and carotenoid pigment. The leucoplasts are the colorless plastids. Amyloplasts store the carbohydrates. The chloroplasts are also double membrane bounded structure which has a membranous sac-like structure called the thylakoid, and the matrix is called the stroma. It is also contain the small double-stranded circular DNA, and the ribosomes is a fat-soluble pigment, keratin and xanthophyll. The ribosome of the chloroplast at the 70 is smaller than the cytoplasmic ribosomes. Thylakoids are arranged in the sacs called the grana. Flat membranous tubules are called as the stroma lamella connecting the thylakoid of the different grana. Stroma contain the required enzymes for the carbohydrates and protein synthesis. Chlorophyll pigment are present in the thylakoid. An elaborate network of the filamentous proteinous structure present in the cytoplasm is collectively referred to as a cytoskeleton. It is involved in the many functions like mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella are the hair-like outer growth of the cell membrane. Flagella are comparatively longer and responsible for the cell movement. The prokaryotic bacteria also possess a flagella, but this is structurally different from of the eukaryotic flagella. The central called the oxoneme. The arrangement of the oxonymal microtubule is referred to as a 9+2 array. Both cilium and flagellum arises from the centriole-like structure which is known as a basal body. They are covered with the plasma membrane. Centrosome is an organelle usually containing the two cylindrical structure called the centrioles. They are surrounded by the amorphous pericentriolar material and lie perpendicular to each other. The central part of the promaximal region of the centriole is proteinous structure called the hub, which is connected with the tubules of the pericentriolar triplet, peripheral triplet, which are 9, and by radical spokes made up of the proteins. Nucleus as an organelle was first discovered by the Robert Brown as earlier in the 1831. Later material of the nucleus was given the name chromatin. Interphase nucleus has a chromatin, nuclear matrix and the nucleolus. Nucleus has a membrane and the space between two membranes is a perinuclear space. Outer membrane usually remains continuous with the endoplasmic reticulum and also bears a ribosomes on it. The nuclear matrix or the nucleoplasma contain nucleolus and chromatin. During different stages of the cell division, cell shows structured chromosomes. Chromatin contain DNA, some basic histone and non-histone proteins and some RNA also. Every chromosome has a primary contraction called the centromere, on the side of which disc-shaped structure called the kinetochore are also present. Based on the position of the centromere, the chromosome can be classified into the 4 types: metacentric, submetacentric, telocentric and acrocentric. Metacentric, where the centromere is present in the middle. Submetacentric, centromere slightly away from the middle. Actrocentric centrosome situated close to the one end, and in the telocentric centromere at the terminal point. Sometimes a few chromosomes have the non-staining secondary contraction at the constant location. This gives a appearance of this small fragment called the satellite. Nucleolus is not a membrane bounded structure. It is a site for the active ribosomal RNA synthesis. Many membrane bound minute vesicles are called as a microbodies, which is present in both plants and animal cells. It contains the various types of the enzymes.